Good morning, students. Today we continue the last chapter, that is reproduction, as already started. In this we continue with the fertilization, or it is also known as syngamy. Before studying this process, we have to remember that is the process gametogenesis and the structure of male and female reproductive system. In the gametogenesis, that is spermatogenesis and oogenesis, we complete or we study the formation of male gamete, that is sperma, and female gamete, that is ova. So let's continue with the next point, that is the fertilization, or it is also known as syngamy. Actually, fertilization process is nothing but it is the fusion of haploid male gamete. Haploid means N. It is denoted as N, that is sperm, and haploid female gamete, which is also denoted as N, that is ovum. Both these haploid gametes fuse with each other and forms diploid zygote. Diploid, that means 2n, n plus n is equal to 2n. So, in the process of fertilization, fusion of sperm and ova, which are haploid, takes place and forms diploid zygote, that is 2n. This process is having much more importance in the life of life of human being, not only in the female but both male and female. Then fertilization actually takes place in the ampulla of the fallopian tube, or it is also known as uterine tube, or it is also known as oviduct, that we already studied in the last lecture, that is the structure of female reproductive system. Then this process, fertilization process, is internal. That developed zygote is converted into or develops into embryo, and embryo develops into the new life, that is fetus. म्हणजे आपला स्वतःचा जेव्हा जन्म होतो तेव्हा पहिली सेल जी तयार होते, दॅट इज नोन ॲज झायगोट. त्या झायगोटची डेव्हलपमेंट होते, त्याचं कन्व्हर्जन एम्ब्रिओमध्ये होतं, आणि हा एम्ब्रिओ नंतर त्याची ग्रोथ होत होत युटेरसमध्ये म्हणजे गर्भाशयामध्ये त्याची ग्रोथ होते आणि त्याचं कन्व्हर्जन फिटसमध्ये होतं. ह्या सगळ्या स्टेप्स आपल्याला 1 by 1 नंतर बघायच्या आहेत. तर आता आपण कंटिन्यू करूयात प्रोसेस ऑफ फर्टिलायझेशन. इन दिस प्रोसेस यू सी दिस डायग्राम. दॅट डायग्राम, आफ्टर स्टडिंग द स्ट्रक्चर, दॅट इज द स्ट्रक्चर ऑफ ovum, that is egg. Last lecture we studied the structure of ovum after the end of oogenesis. See here that ovum is having the structure, that is vesicular body, then polar body, then first outer membrane, that is known as corona radiata. Inner to this corona radiata there is zona pellucida. Inner to this zona pellucida there is vitelline membrane, then vitelline space, then yolk membrane. Actually this is the oocyte which is developed during the oogenesis process. It develops during the second meiotic division. Also simultaneous it forms the first polar body. Actually fertilization process, remember that term is important, that sentence is important. Fertilization takes place during the second meiotic division. That means when development of the first polar body and oocyte. Then see here in this diagram, the number of sperms are attracted, first step of fertilization. That means movement of these sperms towards the egg. It is important. It involves the sperm's capacitation. It is important. Then sperm capacitation into the female genital opening, that is vagina. It is a part of reproductive system, again female reproductive system, and it requires about 5 to 6 hours. Then acrosome of the sperm, during that stage, acrosome of the sperm becomes thin. Calcium ions enters into the sperm for the energy, and the tail shows the whiplash. Remember the term whiplash movement. That means for the movement, energy, the mitochondria are present in the sperm. We already studied in the structure of sperm. There is a row of mitochondria in the middle piece. Thus, the sperm with the movement, whiplash movement, enters towards the egg or oocyte. Thus, the sperms enters through the vagina, that is female genital opening, to the uterus, and then to the oviduct, and then this movement or this structure is observed. The sperms in the oviduct can remain viable for 24 to 48 hours. Then it enters into the egg membrane by releasing, the sperm releases hyaluronidase enzyme. Remember the acrosome of the sperm. Last time we discussed about this. The fertilization releasing membrane किंवा the egg membrane releases, egg membrane ruptures by the acrosome which contains a enzyme hyaluronidase. Also the corona radiata secretes the secretion that is known as anti-fertilizing. That means oocyte secretes the fertilizing, which is a glycoprotein, and sperm head produces anti-fertilizing. Thus here for the rupture or for the entry, the fertilizing and anti-fertilizing reaction takes place, and sperm gets attached to the surface of oocyte. Then it enters, but when the sperm enters in the oocyte, it destroys the sperm outside. Only the head region with nucleus and the centriole gets entered into the oocyte. That means the entry of sperm into the egg membrane takes place. Here the membranes or the secretions, that is the hyaluronic acid आणि zona lysine, zona pellucida also secrete acrosine enzyme or zona lysine, and thus then the entry, the sperm gets entered into the oocyte. Then as soon as the sperm touches this vitelline membrane, it undergoes changes and physicochemical changes takes place here, and the vitelline membrane gets converted into fertilization membrane. हा पॉइंट फार महत्वाचा आहे. फर्टिलायझेशन मेम्ब्रेन इथं जर तयार झालं, फॉर्म झालं, तर नंतर जे स्पर्म इथं अटॅच झालेले आहेत त्यांना एंट्री मिळूच शकत नाही. दॅट मीन्स धिस फर्टिलायझेशन मेम्ब्रेन विच इज फॉर्म हिअर, it prevents the entry of other sperm to avoid polyspermy. That is important.